Hey, everybody. Uh, First, I'd like to welcome Raymond Archambault, or I'm going to mess your name out, Archambault, and his wife, Kit, who are here from Montreal. Raymond is the curator of the CMFF Fungarium at the University of Montreal. And so welcome, Raymond and Kit. Thank you for coming. So we have a surprise guest tonight, a mentor and former professor of our presenter. So it's all yours, Dennis.
My name is Dennis, and I'm a surviving academic. It's my pleasure to introduce tonight's speaker. Brian doesn't know this. Thank you, Michael. I've known Brian for 30 plus years now or so.
You have to stand in front of my computer.
I can do that too.
All right, there you go.
Right. Yeah, so it's my pleasure to introduce Dr. Brian Perry. Brian got his undergraduate degree at San Francisco State and got interested in mycology by taking one of my mycology classes there. And then he stayed on and got his master's degree with me, and he did his master's degree on the genus Mycena from California. Parts of it are published, but most of it isn't published yet. Hopefully we'll get to that at some point. And then he left San Francisco State and went out to Harvard University, where he got his Ph.D. And at Harvard, he studied he moved from Basidiomycetes to Ascomycetes, little groups of cup fungi, and studied the family of Pyronemataceae. You know them as some of the little burn Ascomycetes. After he got his Ph.D., he came back to San Francisco State and did a postdoc in my lab for about 3 years, studying the genus Mycena again. And then he went off and got his first academic job at the University of Hawaii on the Big Island, and was there for a number of years as a professor there. And then he left Hawaii and came back to California and took a position at what we used to call Hayward State, what we all know now as CSU East Bay. So he's been at CSU East Bay now for a number of years. He made the mistake of being the chair of the Department of Biology at East Bay for a number of years, but then he got wise and he stepped down from that. So now he's matriculated back into the department as professor of biology at CSU East Bay. We are hoping that gives him more time to do some research and to do some more collaboration and to collect more fungi and to be more part of the public sphere as well. Being chair is not fun. Being chair in the CSU system is even less than fun. But now he has more time. So tonight he's going to talk about some projects that we have been working on for at least the last 15 years. And these are bioluminescent fungi. So I'll leave it to Brian to tell you all about it and to illuminate all of us into this wonderful topic. Brian?
All right, let me switch this over so the folks at home can hear me. All right, folks at home, can you hear me? Yes. All right. Great. Well, thank you for that introduction, and thank you for the invitation to be here tonight. It's always a pleasure to come and give you folks a talk. I was trying to remember the last time I had been to the Randall Museum to either attend a talk or give a talk, and it was definitely way before the pandemic. So it's nice to be back. As someone said, it hasn't changed at all. It looks the same. But yeah, as Dennis mentioned, what I want to speak about tonight is basically some research that we have been doing for the last 15 plus years. And as Dennis mentioned, after I received my PhD, he and I had written an NSF grant together to get me back out to California, back to the Bay Area where my family was, to do a postdoc with him to work on the systematics and evolution of the mycenoid fungi. And so when I landed back in his lab, back in 2006, right? It was 2006, a long time ago now. He had just published a paper with some colleagues from Brazil and Costa Rica describing bioluminescent species of mushrooms. And it really started this whole sort of boondoggle of research, this whole path and a whole area of research that I never envisioned. I knew a little bit about fungal bioluminescence. I knew that these things existed. I thought they were really cool. but I just happened to be coming back to Dennis's lab at just the right time, and it has taken off. It's probably become the most high profile research that's been done in my lab since I got back to California, and it's been a ton of fun. And so what I want to do tonight is tell you folks basically what do we know about fungal bioluminescence. So illuminating the fungi and illuminating all of us on what we do know and what we don't know about these organisms. And this is a talk this is me right here. I'm Brian Perry. You just heard from Dennis. And then we have two collaborators out of Brazil. We have Cassia Stevani and Anderson Oliveira. And the two of them are biochemists that we have been working with for a while. Sorry, my computer doesn't want to advance for me. All right, there we go. And so we are to have some photos. Here's our collaborators. Dr. Stevani is still at University of Sao Paulo, and Anderson Oliveira is now at Yeshiva University in New York. But we continue to collaborate. And we have published a number of papers. And so this is research that goes back over 15 years when this all started. And we've been publishing, at least maybe kind of like one paper a year, it seems like, since then, mainly we have a huge cohort of really wonderful collaborators. This is the paper that started it all off. So I was just arrived in the lab after this paper was published. So a new bioluminescent agaric from Sao Paulo, Brazil. And this was the first paper that Dennis worked on. Since that time, we've published a handful of additional papers with some of the same collaborators, as well as new collaborators, and even had some wonderful covers of some of our journals with these amazing fungi. And so this has been a project long in the making. And before we get too far into things tonight, I do want to talk about really what is bioluminescence. And importantly, I want to separate it from something that's become really common nowadays is people carrying around their UV lights and looking at fluorescence. Okay, so what we're talking about tonight is bioluminescence, and that is the production of light and the emission of light, which we can measure as photons, by living organisms. So we're going to be talking about chemical reactions that are occurring in the cytoplasms of the cells of these fungi. And I want to distinguish that from fluorescence, right? And fluorescence is simply the absorbance of light and basically the immediate emittance of it at a lower energy wavelength. And this has become something that's really common taxonomically, and many of the field biologists and field mycologists that are out there these days carry around these little 365 nanometer wavelength UV lights, and we can do things like look at some fungi. This is a Mycopan scabripes here, and what you're seeing is this is what it looks like without UV light on it. And then if you basically put a box or a dark towel over it, shine that UV light, you'll see that in many species, we get the illumination of these pigments that we find in different tissues. And it turns out this can be really taxonomically informative for us. So it's becoming a more and more common tool that we are using in the field. And of course, many of us know that many different arthropods also they fluoresce, excuse me. So if you're out in the desert, camping, you know that you can go out at night and look for things like scorpions, other insects, right? And many of these species have the ability to absorb this UV light and re-emit it at a lower wavelength. And it makes them really easy to find. This is one of our Southern Sierra millipedes here. This was taken last year, early in the season, down outside of Fresno, basically. And we were up in a campground at about 8,000 feet. And as soon as the sun went down, we basically got out our UV flashlights and started walking around. And there was literally thousands of these things just crawling all over the ground. But as soon as you turned off your UV flashlight and just use your headlamp or a regular flashlight, they were almost impossible to see. They blended into the forest floor perfectly, but using the UV, you could find them really easily, okay? The other term that you might have heard is phosphorescence, right? So you think of glow sticks. Phosphorescence is another type of reaction where it's absorbing light energy, but unlike fluorescence, it emits it very slowly at a lower wavelength over a longer period of time. So if you think back to when you were a kid and you had those glow-in-the-dark stickers, right, and you expose them to light in your room, and then when you turn off the light, they would glow for hours. That's called phosphorescence, okay? But we, of course, are talking about bioluminescence. So we're talking about chemical reactions going on in the cells of cytoplasm. And we look across the tree of life and see where bioluminescence occurs. It occurs primarily in marine systems, right? So marine systems, we have things like fish, we have jellyfish, we have dinoflagellates. When we get into terrestrial ecosystems, we see insects, some bacteria, as well as, here's like things like glowworms. And then this is another one of our Sierra bioluminescent millipedes. This is another photograph that we took last year. This is from closer down to Los Angeles. One of my visiting researchers went down and collected about 400 of these bioluminescent millipedes in the Sierra Nevada. Unfortunately, all of them perished because we had to grind them up and extract their RNA so that we could basically study the biochemical pathway that leads to this. But these critters are amazing. So if you want to see bioluminescent millipedes, go down to the Southern Sierra in the summer months and go on a moonless night and go out. And these things are super bright. They're pretty easy to find. You can find them during the day and then kind of go back to that location at night and find them. And so when we look at across the tree of life, we see bioluminescence occurring in all these different systems. So in marine systems and terrestrial systems. And in many cases, we know why. I'll get to that in just a minute. But if we look across that tree of life, what we see is that we think bioluminescence has evolved independently more than 40 times if we look across all the different groups that it occurs in. If we look at the biochemical pathways, they're very similar in the chemical reaction, which I'll talk about in just a few minutes, but they're distinct. So if we think about the genes that are responsible for the components of these pathways, we know that they're independently derived, but they're very similar in their overall chemical makeup in the reaction that occurs. It occurs in a very large number of distantly related organisms, over 10,000 species, 800 genera, in 13 different phyla. And so if you look at this tree here, this is from this wonderful resource that's online. Trees from 2010, but it's really going in and it highlights all these different evolutionary lineages where bioluminescence occurs. I know you can't read that where you're sitting, so there's fungi there, okay? So pretty amazing when you think about it. So this phenomenon has evolved independently in widely, variously related groups over 40 times in the evolution of life. And the question, of course, is, well, why? And when we look at certain groups, it's pretty obvious, right? So if we think of the anglerfish here, right, made famous by such fine films as Finding Nemo, we know they have this little appendage up here that they use to attract prey species. Okay, if we think of things like our millipede here, probably using this to find other millipedes, perhaps to attract a mate, right? We think of fireflies. We know that fireflies have a nuptial flight where they glow and they use that for mating purposes. These are glowworms from a cave in New Zealand. These are actually the larval stage of a fungus gnat, and they produce this big mucilaginous sticky net or web, and it's illuminated, and they use that to basically attract and then trap little flying insects that they then consume as a food resource. In the case of jellyfish, they're probably using it as warning coloration in some cases, or perhaps to attract prey, or perhaps even to find mates. We don't know entirely. And then, of course, we get things like dinoflagellates. This is, I think, San Diego in the background right here. And these are dinoflagellates that are being tumbled in the surf, right? And so we think many of these organisms in the marine systems that do this probably do it as a startle response, as a way to basically evade predation by other organisms that are out there looking for them. So sort of a burglar alarm, if you will. They basically glow and disorient these things that might be eating them. So lots of different reasons why these organisms have evolved to bioluminesce, okay? And we get back to fungi, this is really what our research program has been focused on. Number one, we wanted to know how many times did bioluminescence evolve within kingdom fungi? And then, of course, what's it doing? And why are these mushrooms glowing? And why is the mycelium of some of these fungi glowing, okay? So if we go back, we look at early reports of fungal bioluminescence, we go all the way back to Aristotle, right? So Aristotle was really the first one to distinguish glowing wood. So wood that is infected with the mycelium of a bioluminescent species, he distinguished this from glowing wood that was glowing due to being on fire, right? So basically embers versus wood that's glowing because it's infected with mycelium. Then of course we got Pliny the Elder, not that Pliny, but that Pliny, right? And Pliny the Elder was a Roman naturalist. And in his writings, Pliny talks about what he said were phosphorescent mushrooms occurring in trees in the Gallic provinces, right? So he's talking about bioluminescent fungi probably growing in the region, which is today, modern day France or something like that. And so we have these really early reports of bioluminescent fungi. Olaus Magnus talks about wonderful uses of glowing mushrooms and glowing pieces of wood that are infected with fungal mycelium in Scandinavia, not only to find your way at night in the woods during these long, dark winters, but also to go into barns, right? If you think about going into barns, we know that you store things like hay in barns and corn, highly combustible. So if you go in there with a lantern, which is an open flame, it's very dangerous, right? It could burst into flames at any moment, or if you dump that lantern, then things are going to go bad. But you can bring glowing mushrooms or glowing pieces of wood in and use those as a light source. Fernandez and then Ramsbottom both talk about soldiers using luminescent barks in the trenches during various wars. So we have the basically soldiers in the West Indies, but also soldiers in the trenches of World War I and II, putting pieces of glowing wood on their helmets so that they could see each other in the trenches, but the wood isn't bright enough for it to be seen by a sniper who might be, you know, several hundred feet or yards away. Okay, so a way for these individuals to see themselves in a safe manner in the trenches during wartime. And then there's also really cool stories about other ritualistic uses of, you know, glowing fungi and glowing wood from around the globe. We could spend a whole night talking about those as well. A little bit closer to home, we actually have a pretty interesting story. We have the Turtle submarine, which is America's first submarine. This was constructed in 1775 and launched in 1776 during the Revolutionary War. And this submarine was designed to go into harbors on the East Coast, go to the Royal British Navy ships, bore a hole in the bottom of the ships from underwater, insert explosives, and then paddle away really quickly, and then the ship would explode. Okay, that was what this submarine was designed for. It was never successful. It tried to basically blow up a few ships in New York Harbor, never managed to do anything, but then the ship that housed this was eventually sunk by the British Royal Navy. So this is somewhere on the bottom of New York Harbor, or someplace close to it. But if you think about it, here's the operator here. Keep in mind, this is a submarine, so it's going to be completely enclosed. And what were light sources at the time? Candles, lanterns. What would happen to this poor guy in here if you put a candle in there with him? Basically, the candle would consume all the oxygen and he would suffocate. So what did they use for a light source? They used bioluminescent wood. So this is known as foxfire, and this is wood that has been infected with the mycelium or the hyphae of a bioluminescent species of mushroom. Okay, and this is something that we can find here on the West Coast. It seems to be more common in the Southeast, where the term foxfire comes from, but we do see it here. We see it every year for the Mendocino foray. We'll go out and basically find hunks of glowing wood. So that's one that we can see here. So we've made a lot of progress in the last 15 or 20 years that we've been working on this research. We still have a lot to learn. And it wasn't until just a few years ago that we had a pretty solid understanding of the chemical pathway that leads to bioluminescence. So we've known for quite a long time that this phenomenon is what I'll say here is that it's a very similar phenomenon, whether we're talking about fireflies or many other bioluminescent organisms, but it's an NADPH. This is a reducing compound, right? An electron donor and oxygen dependent reaction where we have a enzyme that is generically called a luciferase. And we use the same term to refer to the enzyme in fungi or fireflies, bacteria or dinoflagellates, whatever it may be. And it's going to basically catalyze a reaction with a substrate molecule, which we generally refer to as luciferin. Okay. Now, if we look at this compound here, so in the presence of molecular oxygen in the cytoplasm, this luciferin is going to get catalyzed by luciferase to produce a compound known as oxyluciferin, water, and light. Okay, and so we've known about this pathway for quite a long time. This was elucidated back in the late 60s, and we've been really working on it ever since, trying to figure out what are the genes responsible for forming these molecules that we see in these cells and the enzymes that are responsible. We also know that all fungal taxa emit light pretty much at the same wavelength, somewhere between 520 and 530 nanometers on the range scale there. And that gives us this beautiful sort of yellow-green light. And that's an important one. So keep that in your mind, that all fungi bioluminesce at essentially the same wavelength. Okay. Now, some more collaborators, working with some folks in Russia and our collaborators in Brazil in 2018, they published a pathway, basically a genetically encodable bioluminescent system from fungi. And so these are all biochemists that we work with. I am not a biochemist. I am an evolutionary biologist. So people tell me how much they hate this figure. To me, it's really amazing. What I want to point out is that it's a cyclic figure, right? So this is a genetically encoded system, but it's also something that can continue to operate. It basically cyclic, it refreshes itself. So we can start with a precursor molecule up here. This is caffeic acid. Caffeic acid is a really common metabolite in many of the substrates that these fungi break down as a food resource. So that's something that they're going to have access to. And then we have a series of enzymes that catalyze this reaction. So we're going to have hispidin synthetase up here. It's P. That's going to basically convert conveic acid into this compound called histidine. Another enzyme here is going to convert histidine into that luciferin. It's going to basically, it's going to add a hydroxyl group to it, and we're going to see that we get the compound luciferin. And then LUZ here, this is that enzyme luciferase. This is the fungal luciferase. It's basically going to, in the presence of molecular oxygen, it's going to add that oxygen to luciferin to create this high energy, unstable intermediate molecule, which very quickly decomposes, right? So it's basically going to go from an excited state to a grounded state. And in the process of doing so, it's going to produce carbon dioxide. And for us, the important thing that we're concerned with is light. Okay, so this is the portion of the pathway where the light is being produced. Then we're going to see that this oxyluciferin molecule is going to get basically converted by another enzyme here, pyruvic acid, back into caffeic acid. So as long as that caffeic acid is there, this pathway can just continue to go in these fungi. So unlike fireflies and dinoflagellates, which only bioluminesce for a very short 24/7, right? We do see that there can be some oscillations in the intensity of the light that they produce. These fungi can glow all day and all night, as long as they have access to caffeic acid and molecular oxygen. It's pretty amazing, right? So what do we know so far? Well, we've known for a long time that all bioluminescent fungi are mushroom-forming members of phylum Basidiomycota, and they're all members of class Agaricomycotina. So they're all things that we would all recognize as mushrooms in this room, right? They're also all white spored, right? In the old days, we used to throw all these things into the family Tricholamataceae. That family has been broken up by the Semitists just to make your life more difficult and hard to remember names and genera and stuff like that. But they all used to be treated in this family Tricholamataceae. Now, as you'll see in a few minutes, they occur in 5 separate families and 5 separate lineages. All of them are saprotrophs, meaning that they're out there breaking down dead organic matter, primarily wood, as their food resource. With one exception, we have one species that is a parasite that I'll show you some photos of. And as I said before, in the old days, we used to say occur in 4. Now we say 5 distinct evolutionary lineages because we just discovered one of these new lineages a few months ago. Okay, and you'll get to see that in a few minutes. And so don't run away. Don't be scared, right? This is a phylogenetic tree. This is what's known to as a mega phylogeny. And this is looking at the order Agaricales. So that order of Basidiomycota that we think of as being the mushroom forming order primarily. This is a circle tree. So if you've ever seen a normal phylogeny that's just basically standing up and it kind of looks like, you know, a bunch of branches sticking out to the side, we just took one of those and curled it back on itself to make it hard to read. Okay. And so we do this because it's a real space saving thing. So if you're trying to print out a tree like this in a publication, this would take 10 pages, right? You just have 10 pages of trees or more, which makes it very difficult. So we have something like this. So don't be scared. We're just going to be looking at a tiny little portion of it right up here. So we're just concerned with these lineages where we see fungal bioluminescence occurring. And a little bit hard to see in here on the projector, but all these ones that are highlighted in green, and you can see the names up here, those are our 5 distinct evolutionary lineages that contain bioluminescent species. What that's telling us is look at all these other branches down here, all these other lineages. No bioluminescent fungi are known from these other lineages yet. So it's to tell you that this is a pretty restricted phenomenon within the mushroom forming fungi. We're going to zoom in and we're just going to focus on each of these lineages. And we're going to take a little tour. I'm going to basically show you some examples of each of these lineages, show you lots of pictures of nice bioluminescent mushrooms. Okay. And so we're going to start up here with the Omphalodus lineage. This is a good one for us because it's one that we probably are most likely to see locally as our local best chance of seeing fungal bioluminescence, okay? That's the family, well, it's Omphalodus is the genus we're looking at. We call it the Omphalodus lineage. There's gonna be another 2 genera in here. And there's about 16 species in here that are known to bioluminesce. And the closest one for us is Omphalodus olivascens, right? In my opinion, this should have been the state mushroom. All right. Jerry agrees. This was the contender with the golden chanterelle for the state mushroom. We like it because it's native to California. It's described from the state. It's not edible. That's why people didn't want it out there. It turns out it's actually toxic. You wouldn't want to eat it. So people didn't want a toxic state mushroom, but it glows for goodness sake. right? How awesome would that have been for a state mushroom? But this is Omphaliotis olivaceus. This had a banner year this past season. I saw a lot of this fall. I was out at the Tilden Fair and I got a big group of people to go into the bathroom with a bunch of these mushrooms and lock themselves in there and try and see bioluminescence. They all came running out about 30 seconds later laughing, so I don't think they saw anything. But it's a very common mushroom for us during certain years. Find it growing on hardwoods. It's poisonous to mammals, so you certainly don't want to eat it. And in this case, you'll notice in my slides, I'll tell you whether the mushrooms and the mycelium glow, or sometimes it's just the mycelium, okay? Remember, if you don't know what mycelium is, mushroom is basically, if you take an apple and an apple tree, that mushroom is analogous to the apple. The mycelium is the tree, except it occurs underground or in a decaying log, wherever it is that that mushroom is breaking down. So the mycelium is really the business end of the fungus. The mushroom is just the fruit, if you will. It's just the reproductive structure that the fungus is producing. So when you go out mushroom hunting, you're catching these mushrooms having sex, basically, right? And this is just a nice photo. So Trent Pierce used to be the one that organized the Tilden Fair. Trent has moved to Colorado. He's doing a graduate degree at the University of Colorado at Boulder. But this is this awesome photo that he took. I always love to show it because it's both a light exposure and a dark exposure. So you can see the bioluminescence. You can see all this beautiful green bioluminescence, but you can also see the color of the mushroom there. So just a nice photograph. This was taken in the Berkeley Hills. So within this group, we also have the genus Neonothopanus. These are primarily things that we're going to find in the tropics, right? This is Neonothopanus nambi. In areas of Micronesia where we work, this is referred to as ghost ears. Ghost ears because this is kind of an ear-shaped fungus that grows on the sides of trees in the native forest. The locals there believe that when you die, your spirit goes into the forest. And so these are basically the ears of their ancestors that are growing off the sides of trees. And when you go out at night in the forest, it looks like this. So these are beautiful, really nice, bright mushrooms that we see growing on the sides of trees, common in both the paleo and the neotropics. We've never been able to get this thing in culture. We assume that the mycelium is luminescent. If you have a luminescent mushroom, you typically also have a luminescent mycelium. And we've never tried eating it. We weren't brave enough. So we don't know if this is poisonous, just like its close relative in the genus Omphaliotis. Okay. And then this one is really important for us. This is Neonothopanus gardneri. This is one from Brazil. And it was described first in 1840 from Brazil. And it grows at the base of these palms and these sort of sandy, dry habitats in certain regions of Brazil. When it was first collected in the 1840s, it was never noted that it was bioluminescent. And it was until Dennis and colleagues basically found this thing glowing, brought lots of it back. Dennis discovered that it was actually this thing, first described as Agaricus gardneri, transferred into the genus Neonothopanus later on, actually by them. And it turns out to be a large mushroom, intensely bright bioluminescence. And what's really important for us is that you can find lots of it, right? So this is our collaborator, Kathy Stevani. These are bags of dried mushrooms that they collected in these field sites. So unlike many of the little teeny tiny mycenas I'm going to show you tonight, you can collect pounds and pounds of this thing when it's fruiting. When we do biochemical research, we need to grind up huge amounts of this tissue in order to extract the compounds that are part of that bioluminescent pathway. So this one is really important for us. We could collect huge amounts of it. We can also get it growing in culture. So here you can see, look at that massive fruiting of it at the base of this palm. Almost looks like chanterelles or something growing at the base of this palm. And these are pretty large mushrooms. Here it is in culture, nice and bright. And you can even inoculate bags of palm debris with the mycelium. So you can even grow this in the laboratory and just have huge amounts of the fungus to do these biochemical pathway experiments. So really important one for us in understanding the biochemical pathway of bioluminescent fungi. All right, so let's move on to the next group here is the Armillaria lineage. There's about 14 species that we've described so far, or that we've recognized as being bioluminescent. And of course, here in California and throughout North America, we have a handful of Armillaria species. We oftentimes refer to them as the Armillaria melia complex. So there's a number of species of Armillaria that we've traditionally called Armillaria melia. Probably about a dozen species in North America hiding under this name. We're getting better. We're putting names on many of these things now. All of them are edible, right? So these are the honey mushrooms that people will go out and collect. People find these massive clusters of them, and they typically will eat the stipes. They'll peel the stipes and eat the, the inside of the stipes. Seems like a lot of work to me. I've never eaten them myself. Some of them are saprotrophs. Some of them are also root pathogens. So you've all heard of the humongous fungus, right? We have a couple of those out there. Those are Armillaria species, right? So we know that some of these are the largest and probably oldest organisms on Earth. And it turns out that their mycelium and these structures that they form that we call rhizomorphs, which are basically these melanized, dense clusters of hyphal cells. They form these things called rhizomorphs. This is basically an exploratory organ that will go out looking for resources. And so here you're looking at wood. Someone's peeled back the bark off this wood, and you can see all the mycelium of the fungus. You can see some of the rhizomorphs here. You can see a big cluster of them there. When you put these things in a petri plate and grow them, the mycelium and the young rhizomorphs are bioluminescent, but the mushrooms are not. Okay, so the mushrooms that we have locally for Armillaria, they are not luminescent, but the mycelium of many of our local species is. Okay, this is also the fungus that causes foxfire. So that wood that was used to illuminate the turtle submarine was undoubtedly a hunk of hardwood from the southeastern forests that was infected, basically being broken down by the mycelium of an Armillaria species. And we can find this locally. A lot of folks, for years, we never bothered looking for foxfire here in the West, but it does occur here. During Soma camp or Mendocino forays, people have brought this in the last couple of years. They'll go out and there's a few logs up around that area where the Boy Scout camp, where Soma camp is being held, that are known to have this on there. So every year we find these nice glowing hunks of wood. And so for a long time, we'd give these talks, we say no Armillaria species have bioluminescent mushrooms. It's just the mycelium and the young rhizomorphs. And then Taylor Lockwood, who many of you know or at least know of, was doing some work down in New Zealand. Taylor got really excited about bioluminescence a number of years ago and basically traveled around the world trying to photograph and film bioluminescence. And this is a species from Armillaria, or from New Zealand, Armillaria limonea. And Taylor basically did a time exposure, and sure enough, the basidioms of this thing are luminescent. So this was our first report of basidioms or mushrooms of Armillaria being bioluminescent. And then our collaborators in Brazil sent us these photos not too long after that. And this is another species of Armillaria from Brazil. This is what it looks like when you turn off the lights. You can ignore that there, just showing that where the cap is split here, you get this nice little intense glowing, even though the rest of the cap isn't glowing. But this is a species that we're currently in the process of describing as new, okay, from Brazil. And it turns out there's more than one. There's a handful of bioluminescent species of Armillaria from Brazil, all of which have bioluminescent fruiting bodies. Okay, so lots more work to be done there on that group as well. And then we have the Lucentipes lineage, only 3 species in this lineage. And I love this one because this is really, for me, what sort of kicked off this whole pathway into studying fungal bioluminescence. Right when I joined Dennis's lab, he had just published 2 species, Geronima viridolucens. It was the first report of a bioluminescent species in the genus Geronima, but Dennis had his reservations. He didn't like, it didn't fit any place else. Geronima seemed like the best place to put it, but he had his doubts that it was really a species of Geronima. It's not. It actually falls out someplace else in the evolutionary tree away from that genus. And so for a long time, we've been saying that this represents a new lineage of bioluminescence within the eugaric lineage or within the eugaric tree. Again, this is one from Brazil that we find in Atlantic rainforests. And this one's pretty cool. It's only the lamellae that are bioluminescent, but they're so bright in many cases that they'll illuminate the whole mushroom when you go to photograph it. And then the other one that Dennis described right when I was starting was Mycena lucentipes. And this is another one that Dennis was not happy. It didn't fit any place else in, you know, with basically, it looks like a Mycena, but yeah, there's some characters there that didn't really agree with other things that we consider to be mycenoid. And so Dennis put it here because he had no place else better to put it. And I think it was about the same day or the same week that the cover of Mycology, the journal in which that species was published, came out. I finally got DNA sequence data back from the type specimens of this, was able to walk into Dennis's office and say, not a Mycena. He was right. It definitely goes someplace else. So for a long time, we've been saying that this species and the other species, the Geronima viridolucens, represent 2 unique new genera of bioluminescent fungi. But it turns out, now that we've been sampling more genes and more species, this actually belongs in the genus Mycopan. Okay, so we have data from several different genes that suggest that our Mycena lucentipes actually falls out with Mycopan scabrapy, which is something that we have here. That's actually the photo I showed you that was fluorescing in the beginning of the talk. Turns out this thing is very closely related to that. This is from Brazil. So we haven't moved it in yet, so I put a question mark here. Either Dennis and I will be transferring this into the genus Mycopan unless someone beats us to it. Hopefully not. Okay, we need to do that soon. And this is also a great mushroom because it is the only bioluminescent mushroom to be featured on a US postal stamp. So Taylor Lockwood has wonderful photographs of this species. Taylor's lifelong dream was to get one of his mushroom images on a US postal stamp. When he saw that the US Postal Service was doing a series on bioluminescent life, he submitted his photographs and it got selected. So Mycena lucentipes has actually been on a US postage stamp. Pretty awesome, huh? All right, and now big lineage, the mycenoid lineage. So 5 different lineages of fungi. We've got the Omphaliotis lineage, Armillaria lineage, Lucentipes lineages. All of those have, a few species or maybe up to 20 species. The bulk of all known bioluminescent species of fungi occur within this lineage referred to as the mycenoid lineage, primarily within the genus Mycena, which is what I've been working on ever since I was a wee tot who basically just started working with Dennis as an undergrad. My first fungal love, as I say. And so within the mycenoid fungi, we have a number of different things that you probably didn't think were mycenas, including this fungus here. This is Pinella stypticus. If you know the genus Mycena, doesn't look much like a mycena macroscopically. If you get it under the microscope, it's a mycena. If you look at its DNA, it's a mycena. Okay, so at some point we'll probably be transferring Pinellus, this species, and a number of other species into the genus Mycena. This is basically something that's common worldwide, primarily in the northern hemisphere. It is inedible. It's very bitter, right? So if you try and eat it, it's styptic. So basically, it'll make your mouth pucker up. It's not a pleasant thing to chew on. And what's cool about this one is that the mushrooms and the mycelium glow in eastern North American populations, but not in European populations. And we had never seen it glowing in Western North American populations. And we made the assumption that it probably did not glow here, okay? So we assumed that there was something going on where the pathway that leads to bioluminescence had probably been silenced in the European populations and the Western North American populations. But then I started to hear rumors. People would say, well, Alan Rockefeller, who many of you know, said, no, I think I've seen bioluminescent Pinella stypticus here in California or in Oregon. Just last week, Nathan Warner, who is one of the fundus collectors, if you're familiar with the whole fungal diversity survey project, he emailed me, collected Pinella stypticus locally, and did a timed exposure with his camera. And although it is very weak, it is definitely bioluminescent. So it turns out at least some of our Western North American populations of this mushroom are bioluminescent. It's really cool. And so he was able to, he handed this off to my graduate student. We now have this thing growing in culture. So this will be a fun one for us to sequence the genome of, so we could compare it with the Eastern North American populations to see how they differ genetically. All right, and then lots of really cool mycenas, right? So mycenas, there's lots of different species that are bioluminescent, really beautiful. This is Mycena chlorophyos, probably one of the more widespread species of bioluminescent species of Mycena we have. Common in the Asian tropics and South Pacific on decaying wood. Both the mushrooms and the mycelium glow. And much like Pinella stypticus, I have collected this thing in the South Pacific where it doesn't glow. So I've done a lot of research in Vanuatu. We find this mushroom all the time in Vanuatu, but it doesn't glow. No bioluminescence to it. So something has turned off that biochemical pathway, we think. So this will be another really fun one to study. Mycena lux coli, another tropical species here. This one is pretty neat because it is eaten by nocturnal spiders. So researchers have gone out and they basically videoed and watched this mushroom, and spiders will come at night and visit these glowing mushrooms and feed on them, or perhaps they're feeding on other insects that are also attracted to the mushrooms. So there's an association there with these nocturnal spiders. Another really beautiful species. And then here I promised you I'd show you a parasite. And so we have one Mycena species which is parasitic on coffee. It causes American leaf spot of coffee, a really virulent disease. And this is common throughout South America. It's a virulent pathogen. And what's wild about this mushroom is it produces both sexually by producing what are normal little mushrooms here, but it also produces these special asexual structures, which are bioluminescent. So both the mycelium and these asexual structures are bioluminescent. And so these things are pretty bizarre. So here's a mushroom right here. These are these little structures here called stilboids. And what they are is just a little stalk and then a little cluster of cells at the end. And it's covered with all these sticky hairs. And notice that it sort of disarticulates. And so this thing, the whole thing is designed so that this fungus can spread itself through a coffee plantation really efficiently due to wind and rain and things like that. So these little heads basically break off and wind will blow them around the plantation, or water will wash them off of one leaf and onto another leaf. They stick to it, they grow, and they will infect that tissue. So a really nice way for this fungus to spread throughout different plantations. And this thing is luminescent. So both the mycelium and these little structures are luminescent. So in theory, if you had a coffee plantation that was infected with this fungus, you could go out at night on a moonless night and you should be able to see those coffee plants glowing, at least when this thing is reproducing asexually all over the surface of the leaves, right? Pretty wild. And then finally, probably the most photographed bioluminescent mushroom out there. This thing is all over social media these days. It's having a moment, basically because it has appeared in Hawaii. So every tourist that goes to Hawaii who's into mushrooms is finding these things and photographing it. This is Phylloboletus manipularis. Looks just like a regular Mycena, but rather than having gills, it's got tubes. And really what's going on here, you just have gills. You can almost see the gills there. Then there's little cross walls. So just increasing the surface area where this thing can make its spores. Really, really common in the Asian tropics and the Pacific on decaying wood. Both the mushrooms and the mycelium glow. I've collected this thing all over the place. It was introduced to Hawaii probably maybe 10 years ago, 12 years ago. And now we're seeing it. It was first found on Kauai. Now we're finding it all over Kauai and also on Oahu as well. So it seems to be spreading in Hawaii. Again, it's not a Phylloboletus, it's really a Mycena. So we'll be calling this thing Mycena manipularis. I think they already call it Mycena manipularis on INAT. And there is an older name, yeah. We have to change it, right? So that's a really great one. One of my favorite photos, this was from some field work that Dennis and I were doing. We would find tons of this thing out in the woods. And so you can do things like bring it back to your house where you're staying and use it as a nightlight, or basically a table decoration. So this thing sat, it basically sat in the apartment we were renting all night long, glowing, sitting on this big, nice piece of wood. Okay, so really, really common species, really, really nice. We also have the genus Roridomyces. Many of you have probably seen Roridomyces rorida here in California. This is this little tiny Mycena that has snot all over the stipe. All right, pretty nondescript other than that. Turns out it's got a lot of relatives that live in the tropics, and many of these relatives are bioluminescent. And so if we look at these, what we see in species of Roridomyces is that it's just the base of the stipes that are bioluminescing. What it really is, it's the mycelium of these fungi that are bioluminescing, and that's just where the mycelium is attaching that stipe to the substrate. Okay, so the mushroom itself is not luminescent. It's just the mycelium, which we can see at the base of the stipe. Now, we get Roridomyces rorida here in California, really common. We'd never checked it for bioluminescence. So a couple of years ago up at Soma Camp, one came in and I asked Alan Rockefeller if he would go in and do a timed photograph of it, and he did. And this thing is tiny. This is an acorn, and here's the mushroom right here. And he did it, basically he went into the bathroom at the camp we were at and did a timed exposure of this thing. You can see there's a 220-second exposure with this camera all the way open at maximum sensitivity, photographed it, and then he turned on the lights of the bathroom really quick and closed the shutter. And so you got both a dark exposure and a light exposure together. What you see right here, that is the bioluminescent mycelium of our local Roridomyces. Again, we call this Roridomyces roridus. It's really a Mycena. It was originally described as Mycena rorida, and it'll be going back to that name at some point. And then finally, just a few more because they're awesome. Mycena sylvolucens. This is one that we described from Malaysian Borneo. I found this thing growing in an orangutan sanctuary on Borneo. And it's probably the only known collection of this species in the world, unless someone has gone back to this region of Borneo and collected it. But another really beautiful one. I photographed this one huddling under a desk in an office, trying to basically find a dark spot where I could photograph it. Don't know if the mycelium is luminescent, but we assume it probably is. This one was growing all over the sides of trees in this tropical rainforest. So you can imagine at night, you would just see these glowing mushrooms all the way up the bark of the tree. Really, really amazing. And another one from Brazil, this is Mycena lux eterna. So you might have noticed some of these names. So lux is in there a lot, meaning light. How about lux eterna? This is one of Dennis's names. The eternal light, right? And so this Mycena lux eterna, when you turn off the lights, it does that. So this one is, again, really, really stunning, beautiful species. When you go out into the Atlantic rainforest at night, you can just see the stems, the stipes of these mushrooms glowing really intensely. And then finally, for our last Mycena, this is an undescribed species. This is one that I collected in Vanuatu in the South Pacific. Got it back to camp, waited for it to get dark. I just had a hunch that when you turn off the lights, it would do something. And sure enough, it does. This one also has glowing stems. So this is an undescribed species that I'm currently working on. Again, we only have one collection of this thing that, so far in the world. All right, so that's it for the mycenoid lineages. And then finally, the last one is the Eocyphella lineage. And this is one that we just published with some of our co-authors from Brazil, about 2 months ago? About December, I think we published it. So this one is really neat. This is known only from the Atlantic rainforest of Brazil. These are cyphaloid fungi. So look at your scale bar, that's 500 microns. Okay, so this thing right here, that's 500 microns. Most of these are between 200 and 400 microns in diameter. These are incredibly tiny, and they're just little hollow tubes, basically. There's no gills on the inside. It's just a smooth surface, and that's where all the spores are being produced. But when you see them in the dark, the margins of the, I should also say these are sort of like little upside down funnels. They're basically hanging upside down, so they're open on the underside. And when you look at them at night or in the dark, it's just the margins of these little cups that are glowing. And so this is Eocyphella lucio ursulata. So it means pitcher of light is basically what it is, because it's sort of like a little upside down pitcher. It's just a little cup and just the margin is glowing. And this is the only cyphaloid species that we've documented so far. It's bioluminescent, right? So probably more out there. All right, so now you're probably asking yourself, that's all good and well, but why do they glow? So why are they doing it? Naturalists and mycologists have been asking themselves for a long time, and there's a number of different hypotheses that have been put forth. One of the original ones was that this is done to attract animals that can aid in nocturnal spore dispersal. So by having these glowing mushrooms, you can attract insects to your mushrooms. Those insects or animals are going to eat those mushrooms, crawl, fly, slither away, and then poop out the spores, or maybe they'll get their spores all over their body and then help transport them. So sort of co-opting animals to help with dispersal of spores, like we see plants doing with pollination, right? Or perhaps they attract predators of insects that would come and feed on your mushrooms or feed on your glowing mycelium, right? So these mushrooms want to persist out in the environment for as long as possible so that their spores can mature and get dispersed, right? So maybe by glowing, you can attract predators that will come and eat those insects that would otherwise consume your mushrooms, therefore increasing the probability that your mushrooms survive in the forest. Warning coloration has been put forth, so suggesting that maybe these are glowing to advertise their toxicity to potential things that might eat them. Well, we see this in animals, we see this in plants. We do not see this in fungi, right? So some of these species that I showed you tonight are toxic, but the vast majority of them are not. So the warning coloration hypothesis doesn't really hold up. There's been some nice work done back in the 60s and then repeated again by our collaborators in Brazil just a few years ago that has demonstrated that insects are definitely attracted to these glowing mushrooms, right? So we know insects love light. Just turn on your porch light during the summer, all the bugs are attracted to it. Same thing with these mushrooms. They're certainly attracting insects, but to prove that that is somehow benefiting the fungus is really, really difficult. Okay, so we haven't gotten that far yet. Our research and the working with our biochemist collaborators really suggests that luminescence is actually an energy releasing metabolic byproduct, right? So as I told you before, most of these mushrooms break down dead organic matter, primarily wood. When you break down wood, there's cellulose, but there's also lignin, okay? Lignin, when you break it down, generates peroxidases, basically reactive oxygen species in the cytoplasm. So we think that the bioluminescent pathway is actually an antioxidant pathway that is helping to neutralize some of these potentially damaging compounds that are being produced as a byproduct of their metabolic pathway, basically because of what they're eating. Okay, so that's a kind of a simple, unelegant answer to why they're doing this. But keep in mind, this doesn't mean that this phenomenon hasn't also been co-opted to serve other purposes. Okay, so maybe it is an antioxidant pathway that's helping these fungi, deal with the byproducts of breaking down wood or breaking down leaf litter as a food resource. But it doesn't mean that it hasn't secondarily been selected for because it benefits them in some other way. Okay, and so that's what we're going with. We hope to discover this at some point. All right, so what do we know about? So that's all good and well. I've shown you lots of pictures. You have a good idea of who the bioluminescent fungi are. I've told you why we think they do it. Then the question I want to end with is, well, how many times has this evolved? Okay, and I've given you some hints already. So what do we know? We know that there's about 1.5 million of species of fungi. About 10,000 of those are mushroom forming. There's less than 130 known species of bioluminescent mushrooms, which means that it's less than 0.005% of all fungi that are luminescent. This is an incredibly rare phenomenon in kingdom fungi, not super widespread by any means. Okay, it occurs in 5 distinct evolutionary lineages, all of which luminesce at similar wavelengths. Okay, and some work done in the 60s and more recently, which I'll show you in just a few minutes, the work that we've done with our collaborators really suggests that the luciferin, which is a substrate molecule, and that enzyme that we call luciferase are probably the same for all bioluminescent species of fungi. So this kind of argues for a single origin of fungal bioluminescence. Okay, so when we first got into this project, that was my biggest question was, well, how many times did bioluminescence evolve within kingdom fungi? In the old days, we're talking now 20-ish years ago, you would basically generate a phylogenetic tree like this, and then you would use some really dodgy math to calculate probabilities at various nodes, little spots in the tree here. And so when I first started this project as a postdoc, we generated a molecular phylogeny for the mycenoid fungi here. And then we went in and we calculated the probabilities that the ancestor to these different lineages were bioluminescent. Okay, as you can see here, we got anything from 86% to 93% suggesting that yes, the ancestor here was probably bioluminescent. But keep in mind, the little dots you're seeing there, those all represent bioluminescent species. So sure, if half of the species in that lineage are bioluminescent, there's a big probability that the ancestor was too. And that's just called sampling bias, right? Because we know there's probably many more things in that lineage that I haven't sampled here. So this is a highly questionable, easily biasable way to sort of get at some of these questions. But in the old days, this was the only thing that we could really do. Okay, so we did this. We came up with some probabilities that yes, for the Mycenaceae here, it suggests that there's a probability that the ancestor to all those fungi was probably luminescent, which means that everything you see on the tree there that doesn't have a dot either lost bioluminescence or we just haven't recorded it yet. We haven't observed it yet. Okay, so we did this. The big question, of course, well, what about, since we see bioluminescence occurring in all 4 of these lineages at that time, this would be the most common ancestor to all 4 of those lineages. Probability that that ancestor was bioluminescent was an unequivocal 65%. So really not informative to us at all. So we couldn't say much with this kind of data. Fortunately, biochemists have better data for answering this sort of question. So working with Anderson Oliveira, he came up when he was a graduate student and worked in the lab with Dennis when I was there as a postdoc. And what Anderson did was he came and he looked at the actual substrate molecule and enzyme that was present in different species from different lineages and was able to determine that it appears that there's a single bioluminescent system in kingdom fungi, right? And the way he did this is really elegant, really cool. He went in and he took sample species from each of the 4 lineages. At this time, we didn't know about that last lineage I showed you. We only discovered that 2 months ago. So for years, we thought there was just 4 lineages. And so what Anderson did, he went in and he did a cold extraction and then a hot extraction. And so one extraction would get you the substrate molecule. The other extraction would get you the enzyme. You could mix those 2 extractions together and they would produce light. right? So if I took something like Armillaria melea here and I did a hot extract and a cold extract from it, and I combine those extracts together, they would glow, right? So that's your substrate enzyme or your substrate molecule and your enzyme. And then you have to put other things in there like molecular oxygen and all the other things to make the chemical reaction occur. So what Anderson did was he did cross reactions. So he took the substrate molecule from Armillaria and he crossed it with the enzyme from Mycena or Neonothopanus, Geronima, and Mycena. So he would do cross reactions where you take the substrate molecule from one lineage and combine it with the enzymes from the other lineages and vice versa. He did this in all possible combinations. And then down here we had a control. This is Phyllobletus grassless. This is a non-bioluminescent species. And so what Anderson was able to show is that you can take the substrate molecule or the enzyme from all 4 of these different lineages and mix them. And in every combination, they will produce measurable visible light, right? Really cool. And when you mix them with the control, nothing happens, okay, as you would predict. And so what this data suggests, of course, is that all 4 lineages share the same type of substrate, which is the luciferin, and enzyme, which is a luciferase. And this, of course, argues for the idea that fungal bioluminescence evolved just once in the evolution of kingdom fungi, but it has been many, many times. And so if we go back and we look at this megaphylogeny, that little green dot, which you can just make out there, that is the most recent common ancestor to all 5 of these lineages up here that contain bioluminescent members. So this would be the spot on the tree where bioluminescence most likely evolved. But that means it's been lost many, many times in all these other species. Either that or we just simply haven't observed it occurring in them yet. Okay. All right. And so with that, I'll say thank you for listening and to thank my folks that have contributed specimens and photographs, as well as my funding sources for this research with Dennis and others. And thanks for listening. Question? Nope. So once the tissue is dehydrated, the cytoplasm, once that cytoplasm basically has no more oxygen in it, it's the reaction stops. And you can also extinguish it by submersing them in like alcohol or water and things like that. Yep. Still trying to work that one out. Maybe. Okay. Oh yeah, people can see. All right. I should also show you the last slide. Oh, now he can see me. It's okay. All right, folks. Any other questions? Let me see if there's. Yep, Tom.
Any sequence data on this?
Yes, and so I didn't get into it here because it's not really my research, but the encodable genetic pathway, we know the genes for the whole pathway now.
Any evidence for horizontal transfer from bacteria?
Not yet. Yeah, so it doesn't look like it. We know that there's there's been a handful of gene duplications is what the research suggests, but no tracking of horizontal gene transfer that we've been able to see.
And any evidence for the enzyme where there's not light emission?
So we see that, yeah, so we're getting more and more genomes to compare these things. So looking at luminescent species, and then, close related things that are non-luminescent. So Pinellus would be perfect, looking at Pinellus things that do luminesce, Pinellus things that don't. And what we'll typically see is that one or more of the genes, as part of the pathway, is non-functional. And so it's sometimes it's just completely missing. We can't find it. Yeah. Norm.
So are these luminescent all the time, whether it's light or it's dark?
Yes.
They don't turn on and off, it's just continuous process.
It's continuous. There is a little bit of a circadian rhythm to it. So that some of the work done by our collaborators have shown that there is variation in the intensity of light being output. But as long as they have caffeic acid and as long as they remain moist and oxygen is available, they will continue to luminesce 24/7. Yeah.
So the other thought might be that if this has been lost so many times, then it must be a sort of expensive process.
Ah.
And then if it's an expensive process, it must be valuable to the ones that we take.
Maybe, right? So if you think about it, so the fact that this is such a rare phenomenon, in kingdom fungi. So if we look at look at this thing. This is, this is about, I think there's 3,500 species on this tree. We only see, and I think I've probably got maybe 70 species of bioluminescent fungi in this phylogeny here. It's really rare. So if this was something that was immensely beneficial to fungi, we know that, that if we're thinking of wood rotters, right? Think of this, the sheer number of wood rotters that we see in these other groups, we would predict that maybe bioluminescence would be much more widespread. If you look at where it's kind of hard to see here, and then go back to full screen. If you look at where the most recent common ancestor to all these species is here, it wouldn't take a whole lot. If you lost that functional pathway, just a little ways down the road, you could actually have lost the ability to bioluminesce in the rest of the agaricales, right? So it could have happened a few times where that pathway basically became disrupted due to mutations, or losses, things like that, or it could have happened a whole bunch of times. We're pretty confident that we're not going to find, a bioluminescent entoloma or a bioluminescent bolete or anything like that. But there's probably certainly more within these 5 lineages here. There's undoubtedly many more species that are luminescent, but still really, really rare. So to me, that argues that it might be a nice way to deal with these metabolic byproducts, but it's certainly maybe not the best way. So like you're suggesting, it could be metabolically expensive. So there could have been a benefit to losing it in other species. But again, that would be really, really hard to prove. Yeah. It is. So these are really early diverging lineages. So certainly early on in the evolution of the agaricales, at least, we see the evolution of bioluminescence. And that's just the most recent common ancestor. We don't see, looking at these other groups down here, obviously nothing in the boletes or Clavariaceae or Hygropheraceae. But once we get up to the common ancestor, to the Mycenaceae, and these other groups, that's sort of where it would have to have evolved. Yeah. Yeah, question back.
To you, does that suggest that you just haven't found the right chemical formulation, or do you think there's some sort of symbiosis or something?
Yeah, I should have been more clear. So what I meant is that we didn't have the opportunity to culture it. So a lot of these things, we go do field work in the South Pacific and places like that. We don't often have permits to bring live material back into the US. So we dehydrate all the mushrooms before we leave country. And so everything that we're bringing back into the country is non-viable, dead. You would have to have permits to bring live stuff in, and that's just a whole nother series of hoops to jump through. In the future, I would definitely want to do that if I go back to these places to do this kind of research. But for most of the projects that we were working on to, where we documented these things, we just simply didn't have that ability. Yeah, good question. Yeah, Miko. Mm-hmm. So Phylloboletus is within Mycenaceae. It's a mycenoid fungus. It would, so Phylloboletus would have been right up in here. So that species is one that is within Mycena, right? We have lots and lots of Mycena species, mycenoid fungi that are not luminescent. And so we just chose one that we had that we could grow. So we had it in culture. I think it was
within the other, like what is it? Very, very.
Yep.
It's possible that if you put a light or a camera on that for long enough, to find another lineage.
Possible. And that's why we talked about it's one of these things that this is really it's an exciting time because bioluminescence is it's kind of all over social media. And iNaturalist, as much as I might complain about it at times, is actually really, really useful because folks are going out and they're documenting this, they're taking these timed exposures and they're putting this information out there. So there's absolutely more species than we've accounted for. In our most recent paper, we had 129 species that have been documented in publications. There's at least 5 or 6 more that are on iNaturalist, but these aren't specimens that we've ever looked at ourselves. So I'm sure they're valid, but we just decided, for our publication we're working on right now, where this tree will be to just use those that have been actually published in peer-reviewed articles. But if you go and look on social media on iNaturalist, there's lots of images of other species of bioluminescent mushrooms, mostly mycenas that I don't know. Yeah. So yeah, so it's really hard. It's to find a dark room to do a timed exposure. As you know, you've done it. It's really time consuming. It's very difficult. You have to have a good digital camera. I have a luminometer, which is a fancy little machine that measures light output. So I can just take a little tube and shove a mushroom in it, put it in the machine, and it'll basically throw out a number if that mushroom is producing any light. So my goal would be to try and sample every single mushroom I can. It's just, there's not enough time. So I need to give everyone luminometers and they can all do it. Yes. Oh yes, and so I actually have so this is coming up. So very, very soon, if not already, you will all be able to go out and buy glow-in-the-dark houseplants. So what we've done, so this whole encodable bioluminescent pathway that I showed you, this cyclic pathway, that basically inserted, has been transformed into plant genomes, and these are petunias that you can go and buy at your local garden supply and plant them in your backyard. And at night when you go out, all the plants will be glowing. So travesty, I know. But there's actually there's a lot of research going on using this chemical reaction to produce natural light as well. And so solutions for energy free light sources and things like that. Yeah. So someone's making a whole bunch of money on, on this pathway. The Russians, yes, not us. So we didn't patent any of these things. But yeah, so this is supposed to, we were told this would be available this spring. So keep your eyes peeled for your bioluminescent petunias. Right. 2 questions, all right. Zoom here. No. What's the difference between green mushroom and a blue algae bioluminescence? Wavelength. So totally different. Going to be similar chemical reaction. So it's going to be a luciferase, luciferin. So those are just generic terms for the substrate molecule and the enzyme, but different biochemical pathways that gave rise to it. They've evolved independently of one another. And in the algae, another groups are just seeing that they're bioluminescing at different wavelengths. That was Donald Gray. And then David Porter, with better photosensitive devices, could you see if so-called non-bioluminescent species are producing low-level light? Yeah, and that's the whole idea. So a luminometer is highly sensitive, much more sensitive than a camera sensor will be. But even the modern digital cameras are coming up with more and more sensitive sensors. So they actually do a pretty darn good job. But yeah, you're right. Having the more sensitive device you have, the better your opportunity of resolving this. What we're actually doing also is we can, and I'm not showing any of that evidence here, but our collaborators have gone through and sequenced the genomes of a whole bunch of closely related things that are non-luminescent, right? And so what we can see is that some of the genes that are responsible for the pathway might be in their genomes, but typically a majority of them are missing. Okay, so what we do know is that there absolutely has been evolutionary losses of these genes. So honestly, it'd probably be easier, since sequencing genomes is so cheap nowadays, or really just developing markers that you could go in and amplify just the genes that are responsible for the bioluminescent pathway. You could just develop a series of probes, and then you could just massively sequence all sorts of specimens and see if they have the genes responsible for the pathway. Yeah. All right. Any other questions? All right. So I think my headphones have died here, so hopefully. Oh, yes. Yep. And so when we grow these things in culture, caffeic acid, right? So caffeic acid is this really common metabolite, but it's oftentimes missing in the substrates that we try and grow these mushrooms on. So rye and wheat, those grains that are really common for growing certain types of mushrooms, are really poor in caffeic acid. So we have omphalotous olive acids. We have that growing in the laboratory, and we'll grow it on rye grain or wheat, and it doesn't bioluminesce. but if you add caffeic acid to the substrate, it starts glowing. And so providing that metabolite for the reaction to occur is really great. So I was talking to someone, we were talking about putting getting a squirt bottle of caffeic acid and walking around the woods and just spraying mushrooms to see if they'll start glowing, right? That wouldn't work. But you can add it to the substrate, and that will increase the amount of light that the fungus is generating. You also saw in the photograph that one where there was a circle around that damaged area. So if you know Mycena haematopus, you know that species, it's the bleeding Mycena. So it's beautiful little reddish Mycena, grows on wood. If you damage the cap and then do a timed exposure of it, you might capture bioluminescence. And part of that is that you're exposing the cytoplasm. So you're just making it even easier to see that light that's being produced. So a lot of the photographs you see of Mycena haematopus, it's just where the cap has been damaged, where it's actually glowing. Yeah.
Seems like some of the bioluminescence is not visible by the naked eye.
Yes, and I should have been a little bit sort of glassed over that. But yeah, so much of this is not visible to the human eye. So all the photographs that I showed you in the talk tonight, those are all species that you could see with the naked eye, except maybe that Pinellus stypticus one I showed you. That was one where that was a timed exposure, a really long exposure, and it was actually still very, very faint. All the other ones that I showed you, you can see them with the naked eye. If you've read David Aurora's book, he says, go lock yourself in a closet and bring a sandwich with you, because you're going to be in there a long time and you're going to get really bored. But you have to go into an incredibly dark room, right? So you can't have any light leaking through the doorways or anything like that. And oftentimes you have to sit for 5 minutes to let your eyes to adjust to darkness before you can see the bioluminescence. Okay, and those ones are faint. But I collected Omphaliotis olivascens last year, and literally during the middle of the day, I walked into my bathroom and closed the door and turned off the lights, and I could see it. And there was even light coming under the door. So certain species that we have are incredibly bright. You can see them really easily. Others really, really faint, and you need to be in a perfectly dark room to see them. And then there's many, many species out there that don't bioluminesce at levels that human eyes can detect. And so those are the ones that you have to have either a camera or something like a luminometer to measure them with. Right. Anything else? Yeah, Mike.
If we're done here, are there any other questions?
If one wanted to make a nightlight, are there any high caffeic acid substrates that would be worth looking at?
We usually just spike whatever we're growing it on. Yeah, so you can just buy caffeic acid. And I forget what what concentration we were mixing it at. You probably found that online, but yeah, we basically just using a pipetter, just add it to the substrate, and within a couple hours, the culture started glowing. So it's pretty quick. So so in theory, you could get something like Omphaliotis olivascens or Pinellus stypticus and just have those growing in, mason jars. And then as long as you just, kept them wet and kept spiking them with caffeic acid, you could keep them glowing for quite a while. Don't know if you'd be able to light your room with them, but it'd be pretty cool. Yeah, or just buy a petunia. Just wait, it's gonna be everywhere. Soon we'll have bioluminescent pets, You'll be able to see your Labrador retriever on the floor at night when you get up. That's right, they transformed into a mouse. So you have, there's GFP mice now, there's bioluminescent mice, fish. So it's coming, people. All right, well, thanks everyone.